so these notes again are going to go on page 41. It's a right-hand page if you're doing a notebook. If you're not doing a notebook, then I suggest you take some notes, because we're going to be talking about things that are at a microscopic level, and it does get kind of confusing. And so you might want to revisit this and have some notes to review, especially since this is not in your textbook. Now, there are links in this PowerPoint that take you to videos that I recommend that you watch. A couple of them we'll look at in class. And then the crash course videos don't line up perfectly where I put them, but they line up pretty okay. And then there's always Bozeman Science. And so as I find other good videos, I'll put links up for you guys, either on Google Classroom, if not directly into the PowerPoint. So you're seeing nothing because I have to unfreeze the screen. Ah, there we go. So what we're looking at is the levels of organization of a skeletal muscle. Okay, starting with the gross structure of a muscle, going all the way down to the microscopic structure of a muscle. So first of all, what we call a skeletal muscle is actually, oh gosh, millions of individual cells together. But every time you contract a muscle, you don't necessarily contract it with all of its force, correct? Every time you contract a muscle, do you contract it with all of its possible force? No, right? You can touch something very, very lightly, more firmly, or you can just whap it really hard. And that's what we learn how to recruit. It's called recruitment. How to stimulate either very few muscle fibers to contract or multiple muscle fibers to contract, okay? So right now, this is not up on Google Classroom. So I know some of you like to take notes digitally, and that's fine, but you're not going to find this PowerPoint up yet because it's still a work in progress. And there's a lot of brain research that shows that, you process things better when you write them. Typing's great, but for really being able to process something, to actually go and write it is much better for you, okay? So let's look at the gross structure of a muscle. When we say a muscle, we're talking about a bunch of muscle cells together, and it's kind of like a big cable. You have, I don't know, if you imagine, if you have a ponytail, right? It's one ponytail, but there are thousands and thousands, hundreds of thousands of hairs in that ponytail, right? Imagine you took your hair and you made a whole bunch of little braids, and then you braided those braids together, and then you did one really big braid of all the braids together. You could pull out an individual braid, a group of braids, or you could take the whole huge braid. I don't know if that's a good example. I'm trying. So here we have a skeletal muscle, and I'll use black because I think it's going to stand out the best for those of you who are in the back. So here's a whole skeletal muscle, and it's wrapped with a membrane called the epimysium. Epi means on top of, mysium. Okay, it's the wrapping around the whole muscle. Within that muscle, can you see there are different bundles of individual muscle cells? Okay, and within each one of those bundles, there are bundles within bundles. So if we pull one of these bundles out and we look at it, this is called a muscle fascicle. One of those bundles is called a muscle fascicle. And each muscle fascicle is filled with lots of muscle fibers. Muscle fibers are what we call simply muscle cells. So a muscle fiber is the same thing as a muscle cell. So the epimysium wraps the whole entire muscle. Each fascicle is wrapped with a perimysium. So the whole muscle is wrapped with the epimysium. The fascicle is wrapped with the perimysium.
Muscle fiber.
Okay. And then between the fascicles, you have something called the endomysium. Okay, so the endomysium goes between the fibers. So the perimysium, if I switch colors here, the perimysium goes around the outside. And then I'll go to pink. And the endomysium goes in between. Doesn't stand out very well.
Wait, how about the Sarcolemma?
I haven't talked about it yet. I'm going to talk about it now. Okay, so now you see this other word here, sarcolemma. Okay, come on, iPad, don't freeze up on me. Okay, the sarcolemma. The sarcolemma is the fancy term for plasmalemma. Okay, and let me erase that and fix it. Oh no, I don't want to erase all strokes. Do you remember what the plasmalemma is? No. Do you remember what the cell membrane is?
Yes.
Okay, the cell membrane is the same thing as the plasmalemma. And the plasmalemma is the same thing as a sarcolemma, only it's specifically talking about a muscle cell. Okay? If I said neurolemma, what would I be talking about?
A nerve cell.
A nerve cell's membrane. Okay? So the plasmalemma is a general term for all cell membranes. If I say neurolemma, I'm talking about a cell membrane on a nerve cell. If I say sarcolemma, I'm talking about a cell membrane on a muscle cell. Okay? Are you guys lost? Okay. So we're talking about levels of organization. We're going from biggest to smallest, from gross to microscopic. So the gross anatomy, the whole muscle as a whole is wrapped in the epimysium. Okay, it's the packaging that wraps around the whole muscle altogether. If I pull out one of the bundles of cells called a fascicle, there are multiple fascicles in each muscle. Do you see how there's multiple fascicles in that picture? Multiple fascicles. Each fascicle is individually wrapped. Okay? So the muscle has a wrapping. Then there are bundles within the muscle called fascicles, bundles of cells, and each of those fascicles has an individual wrapping called the perimysium. The fascicle itself is made up of lots and lots of muscle fibers, which are muscle cells. And there's wrapping in between the muscle cells called endomysium. So it's like layers of packaging. Each muscle cell, it's a cell. It has to have a cell membrane. It's got a fancy name, sarcolemma. That's the fancy name for the plasma membrane. Okay? Are you unlost now? Have you been found?
Kind of.
Kind of? Okay. So you're going to have to look at this more than once. And I think this is something that you drew, isn't it? One of your left-hand pages. So draw it, and don't just draw it, but make sure you understand it, okay? It's layers of organization and packaging. Now, when we look at the muscle fiber itself, it's got all these little contractile units. So you see where it's got something pulled out? Those are the contractile units. That's where the actual shortening of the muscle occurs. So there are multiple contractile units. And then there are also these little organelles here, and there's another one there, and another one there. And those are mitochondria. What's the job of a mitochondria?
Energy source.
Okay, it's the powerhouse of the cell. It makes energy in the form of ATP from the fuel that we know as glucose, sugar. Excellent, excellent. Okay? So if you have lots of mitochondria, then that means that this cell must have a high ATP need. And two things are super important for muscle contraction: ATP and calcium. Yes?
So where's the mitochondria located?
Okay, so this whole thing is, let me see if I can draw you a picture. Okay, so, that's the cross-section of a muscle fiber. And each one of these circles represents a contractile unit, the actin and myosin proteins. And in between them are mitochondria. See, because this is the cell membrane, also known as the sarcolemma. And in there somewhere is, guess what? A nucleus, because it's a cell. And this is cytoplasm. And in the cytoplasm, you have to have all of the equipment that a cell needs. So you're going to have mitochondria and Golgi and ribosomes and all that stuff, right? But we're not showing all that other stuff because right now we're focusing on the part that makes it contract and do its thing. So we are paying attention to mitochondria and these contractile units, these bundles of proteins. Got it? Hey, what's the name of the cell membrane on a muscle cell?
Sarcolemma.
Sarcolemma. Sarcolemma. It's on there. Okay? Sarcolemma. This is never going to really go away. As long as YouTube exists, it'll be there. That's a frightening thought. Okay, I'm going on. So now I want you to have this introduction so you understand the level, the scale that we're looking at. See, these little words just aren't enough to explain. So those bundles of fibers that I were talking about, these are our fibers that we're going to be looking at. Thick fibers are myosin. Thin fibers are actin. So that's what I want you to write down. Thick equals myosin. Thin equals actin.
That's what I did.
Okay.
That's all I remember I took notes on is actin.
So that's basically, your book kind of leaves it there. Doesn't really go into a lot of detail. We're going to go into a lot more detail. This is probably going to be the most detail-rich lecture on physiology that we've had the whole semester. Because naming muscles is all good and fun, but I think the real amazing part with the muscular system is that they're little tiny machines and how they make our muscles go, how they make our muscles contract. It's really quite amazing. So, these are proteins. And proteins are sensitive to what? Heat. Heat. And pH. So, this is what happens why meat changes when you cook it or you expose it to acid. This is why shrimp tails curl and become that orange and white color when you heat them. That curling of a shrimp tail or a lobster tail when you're cooking it is the contraction of the actin and the myosin filaments because of the heat. They're chemically, those proteins are chemically changing when you add heat. And you can't uncook them. And so that's why they curl, because that's contraction. And if you curl it, cook it too much, then they get really tough. Yeah? Unless you know what I'm talking about, unless you're allergic to shrimp. Which is bad. Then you don't eat shrimp. Because it can kill you. Okay, so this picture is just another diagram showing you the gross anatomy of a muscle connected to a bone by a tendon, and then pulling out the little fascicles wrapped with the white perimysium, and then pulling out an individual muscle fiber. And it shows you here on this muscle fiber, here is the plasma membrane, the sarcolemma. So now we're here at a cross-section of an individual cell. And out of that, we get these contractile units. And these contractile units, this is what causes skeletal muscle to have its banded pattern, its striations, because of the way the actin and the myosin are arranged with each other. Remember, actin is the thin filament, myosin is the thick filament. Okay? Actin is thin, myosin is thick. Let's see, how am I going to remember that? Because I always have a hard time remembering that. Okay, A comes in the alphabet first, so we all start out thin. Ah.
A.
Ah, right? And as you get older, if you're not careful, you get thick, don't you? See, M comes later in the alphabet. A is, now I'm never going to forget it, huh? I believe it or not, I started out then, ladies and gentlemen.
If you're drunk, you don't know the alphabet. Oh.
Okay, well, I think if you're drunk, you're probably not talking about muscle contractions anyways. Okay. So, actin is the thin, myosin is the thick. Now, you drew something like this for a left-hand page. The key to muscle contraction, without knowing anything about how it happens, is you see this zigzaggy line. This is called the Z-band. And what happens is the length of the muscle fibers don't change, but they slide over each other, allowing the Z-bands to get closer to each other. And so I like to use my hands to do this, where I take my hands, thumbs up, and my fingers represent the thick and the thin filaments. And as the degree of overlap increases between my fingers, the thick and thin filaments, my thumbs get closer together. So if my thumbs represent my Z line, my muscle here is contracted. When they go farther away, then it's relaxed. See, contracted, overlap, relaxed, less overlap. Now, they're always overlapping somewhat, but maximal contraction is when you have that maximal overlap between the thin actin filaments and the thick myosin. And this is called the sliding filament model. Oh, I'm doing so much better this period. Okay, I'll get there. Okay, so the degree of contraction is a result of how much overlap you have. And the process of overlap is called the sliding filament model. What filaments are we talking about? Actin and myosin. Okay, which one is the thick one? Myosin. We are going to remember that now, aren't we? Okay. So actin is the thin filament. And I have to tell you guys, it wasn't until I just thought of that. I always have a hard time remembering those. Now I won't. Which one is the thick and which one is the thin?
Actors are thin and actin is like,
Okay. You can, if that helps you to remember it, but I like I started out thin and now I'm thick. Actin is first. Okay? So, the thick and the thin filaments slide past each other as a muscle contracts. So the myosin slides past the actin. So if you have a greater degree of overlap, you have a greater degree of contraction. And that would make the Z lines close together.
So what you said, more overlap, more contraction, Z lines go together?
Uh-huh.
Okay.
The Z lines moving closer to each other is a sign that you have more contraction. Question?
And the overlap is like the myofilament.
Uh-huh, the myofilaments, the actin and the myosin overlapping each other. Okay? Oh, I'm doing so much better this period now. Well, I told fifth period this is not what I was talking about. I was just kind of disjointed and it just didn't flow. And I told them I wasn't going to record fifth period's lecture because I wanted to be, have it flow really nicely. Okay, so the writing's going to go away. Are you guys okay?
No.
Okay. So the two things that are required to allow those Z bands to get closer to each other, calcium and ATP. And I hope that you remember that ATP can be turned into ADP plus a phosphate and energy is released. But if you add energy in, you can take ADP plus phosphate and turn it back into ATP. Do you remember that process of phosphorylation?
Here, let me draw that.
So ATP has higher energy, higher potential energy than ADP.
Here, let me write this down.
I'm waiting.
I have bad memory.
Okay. All right. Where did the energy come from to take ADP to glue on that phosphate onto the ADP? Right? Cellular respiration.
Oh. Wait, it takes glucose and cellular respiration?
Yeah, glucose and the process of cellular respiration and to convert the chemical energy that's held in the glucose molecule into the chemical bond energy that's held in ATP. That's what cellular respiration was all about. So it's just energy. The first law of thermodynamics, energy cannot be created or destroyed, but it can be transferred. So we're just talking about transferring energy from glucose molecules to ATP molecules. And now we're going to look at how the energy from that ATP molecule gets converted and transferred to muscle contraction. But every time you transfer, it's not 100% efficient. And this is why our muscles create heat, because that energy transfer is not 100% efficient. And heat is always lost each step of the way.
Wait, does the ATP energy stuff will keep you up? Because somehow I confess I fell asleep in computer class, and my teacher's like, Amber, you want some Honey Nut Cheerios? I'm like, oh, okay, Honey Nut Cheerios.
Well, we need to look at what we're eating for lunch, because if you eat a lot of carbohydrates at lunch, it raises your blood sugar up really quickly, and then you have this blood sugar drop right after lunch.
I don't eat as much at breakfast. And
So you need to eat good protein breakfast and lunch. What's breakfast? Okay, we're not even going to go there. All right, here is a more simplified diagram, just showing you the locations and the relationships between actin, your thin filament, and myosin, your thick filament. It is the actin molecules that are going to create, and almost it's like, can you imagine like a tug of war, where you're pulling the rope closer, closer? The actin is going to be pulling on the myosin. Okay, my bad. Backwards. The myosin is going to be pulling on the actin,
Okay.
pulling the Z lines closer together. The myosin pulls on the actin. So we'll look at how that happens. That happens when cross bridges are made between the myosin filaments and the actin. So you see these little knobby ends here? Okay? Those knobby ends connect to myosin binding sites on the actin, and they do this power stroke. They grab and turn, which you see in the bottom diagram, the contracted muscle, there's more overlap between the actin and the myosin. And that's because, is the actin changing length? No, and the myosin is definitely not changing length, but they're overlapping more. So the distance between the two Z lines has decreased. But the length of the proteins doesn't change at all. So overall, when you have all of these contractile units shortening, it shortens the entire length of the muscle, which then pulls on a tendon, which moves a bone and causes flexion or extension. And that's how we move. But still, we haven't gotten to why and how. So now we have to talk about something called the neuromuscular junction. Okay, neuromuscular junction. Sounds all complicated. What does neuro mean?
Those nerves.
Nerve. Muscular refers to. And a junction is just a place where two things meet. So, conjunction, junction, what's that function?
Oh, God.
Okay, so, okay, Schoolhouse Rock, I know. Well, that was on when I was a kid, though. So, the neuromuscular junction is the point where a nerve ending, okay, comes in very, very, very close contact with the muscle cells, but they don't actually touch. So, here we have one nerve, and it's neuromuscular junction. The neuromuscular junction is all of the myofibrils that are being innervated and touched. So one nerve may touch 3 or 4 different muscle cells. Here you're only showing one muscle cell being touched. But this nerve might have another axon over here that touches another muscle cell, and it might have another axon over here that touches another muscle cell. So when this nerve fires its impulse, it's going to be stimulating those 3 muscle cells. And what do you think is going to happen if you have lots of nerves being stimulated? A stronger contraction. And that's called recruitment. So your brain is pretty amazing. It looks at an object like this little purple paper clip here, and it says, oh, that's pretty small. It's going to be pretty light. I don't need a lot of neuromuscular junctions to be recruited. I don't need a lot of muscle cells to pick this up. Whereas the stapler, it's bigger, and I look at it, and I think it's made out of metal, so I'm assuming it's going to be heavier. So I need more force to pick it up, so I must be recruiting more muscle cells to pick this up. And then I'm going to look at this book, and I'm going to pick it up, and I'm going to go, oh, man, I might even need 2 hands because I need to recruit even more muscle cells. But how about that Amazon box on my doorstep when I get home today, and it's absolutely huge, and I assume that by its size, it's going to be super, super heavy. So I've been down to pick it up, and little do I know that it's just been mispackaged, and it's just one thumb drive inside of it and a whole bunch of styrofoam peanuts. What's going to happen when I pick it up? I'm going to almost throw it into the air, right? Because my brain said, oh, you're going to need a lot of muscle cells to pick up that object. And so it recruited all these muscle cells, but then you pick it up and you're like, whoa, that was a lot lighter than you expected to be. What about if the opposite was true? If it was a small box but filled with lead? I assume that it's going to be pretty light. I go to pick it up and I go, whoa, that was heavier than I thought. And then you stop, and then your brain automatically adjusts and recruits more muscle cells, and then you pick it up, and you can pick it up the second time. You guys have all experienced this. Have you ever seen a little kid pet a dog or a cat the first time? and they like whap the crap out of it. They're like, nice kitty, and the cat's like, ah, you know. And you have to teach them soft. You don't say you need to recruit fewer muscle cells when you touch the cat. But that's literally.
That's why my dog is afraid of kids.
Yes.
Well, because I'm not used around kids, and the kids are.
Yes, and kids don't know their own strength, right? The kids don't know their own strength, and so they have to learn not to contract as many muscle cells. So that's recruitment, another concept that is in your textbook. So, at this neuromuscular junction, you see the little synaptic vesicles. These are little packets of what we call a neurotransmitter. And so I'm going to erase this word here, and I'm going to write the new word, neurotransmitter. Neurotransmitters are chemicals. Neuro for nerve, transmit, meaning to send a signal. Because nerves don't actually touch each other, they release chemicals that stimulate the cell they're trying to contact. Neurotransmitters are the chemicals that are going to be released by the nerve from these little vesicles, and they float across the space called the synaptic cleft, because a cleft is a fancy term for a space, a gap. And they fuse with the cell membrane of the cell that they want to stimulate. In this case, guess what? It's the sarcolemma of a muscle cell. The name of the neurotransmitter that we're going to be talking about is acetylcholine. One of the very, very, very important neurotransmitters. And we abbreviate it ACH. So acetylcholine is the chemical that tells nerve cells you're gonna contract. I told you this is the most complicated lecture that we've had so far this year.
Wait, the school year or the year 2017?
Amber, this school year in my anatomy class.
Oh.
I know that by far this class is much more difficult than you probably expected it to be. But look how you've risen to the challenge. Okay? Yes?
It's nerves and nerve cells to contract?
No.
Oh,
Muscle cells. Yes. Okay? That acetylcholine is going to tell the muscle cells to contract. So here we go. Getting more and more complicated. This doesn't actually show the acetylcholine, but it shows that you have the cell, the contractile cell, which is the muscle. Okay? And there are these deep, this is the muscle cell here. This is all muscle cell, the cytoplasm of the muscle cell. And inside the cytoplasm of the muscle cell are your thick and your thin filaments, actin and myosin. And you have the plasma membrane, also known as the sarcolemma, which I outlined in black. And then you have this structure here called Sarcoplasmic Reticulum. Oh my gosh. Another word. It's absolutely like an endoplasmic reticulum. In fact, it's the same thing. I can spell, really I can. Reticculum. And that equals the endoplasmic reticulum. The only difference here is we're talking about ER that's specifically in muscle cells, and muscle cells have a ton of it. More than any other cell, okay? They have a ton of this tube network. And inside of the sarcoplasmic reticulum is calcium. Calcium ions, and lots of them. So, when the acetylcholine is released, the acetylcholine bonds to the membrane of the muscle cell, and it opens channels in the muscle cell. that creates a difference in electrical charge. Makes it negative outside and positive inside, I would think. Okay? It depolarizes the membrane. Okay? And that then, that charge is transferred deep into the cell through these things called the T tubules, which then releases more calcium out of the sarcoplasmic reticulum. So you already had a lot of calcium, but then a whole bunch more gets released. And that calcium then goes to the next stage. So I want to stop and show you a video really quick. Okay. I have to go out of writing and up, 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 go back. And I have no sound. This is not good. Where's my sound? Where's my sound? No, it's not playing the sound.
You use muscles. There we go. A muscle can produce enough force to move the body.
Sorry, gotta go back to the beginning. There we go.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Nice.
Okay, so I think that's like absolutely a fabulous video. I can't say it any better myself. So there's a video embedded there, and then to help us tomorrow, nice easy steps for contraction and relaxation, a diagram, and 2 other YouTube videos that you can watch. This second one down here on the bottom, the second one on the bottom there is, I think it's the crash course part 1. Okay? So I know this was long, especially because we added that video, but much better communicated than I did in 5th period. So, thank you.